Most of the colors, Hugin, are fluorescent colors, and so the fluorophores, which are the molecules that glow at a particular wavelength of light, tend to be either red or dark red, green, or yellow. Blue is very rare because it's a lower wavelength of light. It's 405 nanometer wavelength of light versus like green is 488 and then 535 or 655 for the reds. The yellows are close to the greens, like also like 500-ish. The purples are even lower wavelength past the 400s, and those tend not to be utilized because they're not as high energy wavelength as those higher ones, like the deep reds and the greens, if that makes sense. They just don't use the purple ones, Hugin, because they're not as effective and efficient. So now he's just saying is that there's different lengths of on your plate of DNA that you can get your sequences to bind to. Sometimes the length can be the exact same, and so then you might have difficulty telling apart which strands are which and how they're binding. But you can modify that with the different salt concentrations in the liquid or different amounts of electricity, and so you can affect how, like, the binding affinities of what's more likely to bind, and so then you can actually parse out what your DNA looks like on this microarray plate. And here is just like this is like the microarray data that you would actually be able to get out of one of these like little plates. There's different resolutions that you can buy. Basically, it's a question of price and just how sensitive they are. So you can go from this low resolution that can tell you, okay, where's the blue gene and where's the red gene, and then along this x-axis is the chromosomal position. So where exactly is the event where the chromosomes are like crossing over and shifting their genetic information? Where's that break point? With the higher resolution, you can actually get like much closer to the actual like chromosomal arm position. And with that, you can figure out, okay, where on the chromosome arm is this crossing over event taking place? And then you can also predict what the DNA structure is like at that particular stage. So if you're seeing crossing over events happening at a particular site a lot, but the chromosomes wouldn't necessarily line up if they're side by side, but it would only line up if it was like some kind of weird curve, then you can predict some kind of positioning of the DNA inside the cell. That's not the end-all experiment for how to like decide how chromosome positions look different inside the cell, but it's just something that we can use as a tool. But this is that red and the white gene that he is referring. And again, on the x-axis is the position of where on the chromosome we are. And then the y-axis is which it's more like. So, like if there's a crossing over event, so everything's the same for a while. And then the split point is when you're having that crossover of switching genes from the red or orange to the blue side of things. And so that flip point, you can see it's actually a total mirror image. So the red and the white swapped positions, and that represents that crossover event. And that's the two genes that they were looking for. Big region he's pointing out is right here. This is now blue and this is orange, and then after that it goes orange and just blue. So there's that one little piece where you switch between the genomes, and that also means like one copy will have one chromosome has an extra copy and the other chromosome has no copies. So this is just a higher res look at it again, and you can see now where that switch point happens. So everything, like if you zoom in here, we have blues and oranges, but they're flip-flopped from the positions they should be. So it's just an extra way of visualizing when that crossing over event happens. Remember, two chromosomes here. This one's blue, this one's orange, and now there's an event where they switch genetic information. You have orange on one that was normally all blue, and you have blue information that's on one that's normally all orange. And so that is a phenomena that we have in our bodies, you know, during development, during reproduction. And so it's just a little extra. It's a cool way of learning and studying how that fundamentally happens. Let's continue on. So this is the technique to look at whole genome recombination rates. And so what you have to do is you put the two chromosomes into one particular organism, one particular yeast. So you have alpha and A yeast. You mate them to make them diploid. You place them on a dish, and then you mutate them. So what's interesting about this part is, too, they do... he's mutating the entire line, like, of the genome, and you don't know where it's going to be mutating. So to actually detect where the mutation is, you have to put the whole lines into the sequencer. So he actually was able to ask if it's mitosis or meiosis. So by just removing one particular gene to prevent meiosis from happening, that's during sexual cell selection, sexual division. By removing that gene, you get rid of that variable, and so you're only looking at mitosis and the effects of mutation on mitosis. So it's just a really, really cool, like, extra bit. So he's just going through now what all you might see on the sequencing. So here's the 2 copies, one blue chromosome, one red. There could be a mutation on one of the chromosomes. There could be losses of chromosomes, so like an entire part of the arm, or a duplication, meaning a doublet. So like one region could be 2 times the amount that it should be. A crossover event like this, so you're no longer totally heterozygous because you've moved some of the blue over to the red, or vice versa. Chromosomal rearrangements, so either part of the chromosome breaks and moves elsewhere, it moves and turns around and sticks itself back onto the original arm, or any combination thereof, or a total loss or gain of a chromosome. So these are all the alterations that he expected just from doing this mutagenesis. And you can do mutagenesis a couple of different ways. There's chemical mutagenesis, that you just put a certain chemical onto the chromosomes and see what happens. There's also UV light. I actually did that experiment on yeast cells in undergrad. Unfortunately, it was a Bio 141 class, and the experiment didn't work because we just cooked our cells. And so that's the unfortunate side of like Bio 141 labs, is that when an experiment doesn't work, then the next week you just continue on like it did, and in your like final paper you just wrote like, oh, what did you think? Why did you think it didn't work? And it's like, it didn't work because I left it in the UV too long. Here's the rate breakdown of what he found. So most of the things that they saw was the single mutations that they were able to pull out. The rest of the majority was they saw crossover events. So this LOH just means loss of heterozygosity, meaning that the blue chromosome and the red chromosome no longer are just blue and red, but now the blue has a little bit of red, or the red has a little bit of blue. And then they saw really, really few instances of chromosome duplication or loss, because that leads to lethality, or large chromosome loss. So you don't lose the entire chromosome, you just lose like a large chunk of it. And so all of those collectively were what they found as like the breakdown events to it, which surprising that you only see these more subtle changes, like a little mutation here and there, or like crossing over events. And that's just because it's less likely to be lethal and to kill the organism. An interesting little aside, he's suggesting that maybe the reason they didn't find so many chromosomal defects was because losing a chromosome or gaining a chromosome slows down the yeast development. And so when you're growing a ton of yeast cells on a dish, the ones that grow better are more likely to grow, and you just won't be able to detect as many. They'll outcompete the ones that aren't growing successfully, which I think he's suggesting is a limitation of the study. You're only selecting for things and picking out things that grow well versus that don't grow so well, right? Because if everything grew at the same rate, maybe these ratios would come out a little bit different. So that's a really nice way of also pointing out an experimental, well, shortcoming, or like some small flavor of deficit, which I think is really neat, and it's really nice to see people say it too. Yeah, so so CHOP, it's a fungus, right? Because yeast is just a fungus. So while they are different from plants, they have a little bit more in common. What was the method of manipulation used here, and what different method have caused different ratios? So Cliff, I think part of it, so to answer your second question is, would it cause different ratios? I don't think so, just because of how they were doing the experiment. So they were growing them on plates, and so even though you've mutagenized them, and so in this one they actually used I think this one was UV mutagenesis, because that's like the old school way of doing it. It works, but you're still you're picking colonies at the end of the day that are growing on your dish, on your petri dish, like agar or whatever on there. Are they growing yeast colonies? And some are going to grow better than others. So even though if you mutagenize in a different way, you're probably going to get the same ratios, not necessarily because that's what actually happens, but it just could be a shortcoming of how the experiment is set up, because you're selecting for growth. If you're making on a big enough plate where it wasn't just selecting for growth, but you're maybe plating a lot more, you might get some different ratios from that. Or just when you plate it, make it when you're streaking the plate, which actually I can show you what that looks like. So it looks like this. So those are like the colonies that you'll get on these plates. From the original source, you'd want to get, like, put it on a stick, and then he's gonna rub on this new plate. If you rub less heavily, you're then more likely to get those slower growing organisms. Ratios reflect the viability or lethality. They do, Cliff, but they also reflect the side effect of the growth rate. So you could be getting just far fewer colonies with the chromosomal inversions just because they grow slower, and the ones without the chromosomal inversions grow more rapidly, so then you just don't get those colonies as like commonly. So it could be just a density question, Cliff. My suspicion is that if using like a chemical mutagenesis, you'd probably get these same ratios. So like, see how he's rubbing? If you just put a small amount on the plate, you might be able to get different growth. But how they did this, Cliff, they took yeast cells and irradiated them, and then were plating. And the way you plate it is actually really, really cool. You take a little bit of sarin wrap, and you put it into the plate, and then you stamp it on a new plate, and then you stamp that on a new plate, and you keep stamping it, so you're diluting it a little further and further. But it is not... You're likely to, like, you're not starting with a pool of the same amount of, like, ratios, right? It just depends on where it's getting hit, and then also the question of how long do you leave them to grow before you do the sampling for the sequencing. Because if you take them too early, they will not have died yet, because you haven't undergone enough cell division to have gotten that effect of, like, what is lethal and what isn't lethal. So there's just a lot of extra factors to consider with these experiments. Like, there's limitations on their front for sure. But I think the ratio is probably more or less the representative of lethality as well. If you also, there's been the argument too of growth rate being a comparable measurement to, like, cell viability. So the more cell viable you are and the better they do in terms of growth, the more likely they are to be viable. So even if you took one of these aneuploid cells in yeast that had more than one copy of a chromosome, or only had one copy instead of two, if they just grow really, really slow, eventually what happens is, if they grow too slow, the dish, like, they're just not able to consume enough, and it'll get contaminated, and they just end up dying. So then their viability then, for another reason or another, is considered as really low. But yeah, again, how much you place on a dish, like, this kind of streaking is more normal, like doing that whole line. And so right now he's doing that whole line on a couple of different zones of the same plate. So see, he's taking, and it's diluting each time of your colonies. But it makes it harder, and so that's what it looks like at the end. It makes it harder because when you irradiate an entire plate, it's not all the same genetic background. Like each cell now no longer is not going to be the same, because they could all be hit differently. And so when you're doing these different densities, you can have tons of different effects. Let me know if that makes any sense at all, Cliff. So this is something that's known now, but this is a fundamental piece of biology that was unknown to many, many people And what it is where is mutation more likely to happen on a chromosome? Not just the yeast chromosome, but any animal chromosome. Regions called the centromere, which is the center of the chromosome, seems to be more protected from mutation than everywhere out. So the centromere, you can actually, while this is a super tiny graph, right, we can actually pick out where the centromeres are. These regions where you don't see those peaks, where you're not seeing, because the peaks show mutation level, those little troughs in the graph are where the centromeres are. And it's just because the DNA is so tightly packed in those regions that for some reason they're just less susceptible to mutation. It's a fundamental piece of biology that was discovered by using yeast, and it turns out this is actually conserved from other organisms as well. For example, having in mammalian cells and insect cells, it's all conserved and kept, which is, I think, super, super cool.
Thanks for watching SciAnts Media, and be sure to hit those like and subscribe buttons to help our channel grow and support our science outreach and education goals. We can be found on Twitch most days of the week, and be sure to join us here for our next video. Thanks again, and stay curious, everyone.